Hello, my name is Thim, and today I will be presenting how partial thermal ablation of melanoma shifts intratumoral IL-1 beta mRNA and immune cell protein levels and patterns. First, a brief background on melanoma. Melanoma has approximately 100,000 new cases per year, where the 5-year median survival of metastatic melanoma is around 30%. Standard of care therapies include surgical resection of the primary tumor site, radiation, chemotherapy like gemcitabine or cisplatin, and immunotherapy like anti-PD-1. Even with these therapies, clinically we have a problem of the responder-non-responder paradigm. One contributing factor to this responder-non-responder paradigm is the tumor immune microenvironment, i.e., cold versus hot tumors. Some tumors are highly infiltrated with T cells or other anti-tumor cells, and those patients tend to have better outcomes when treated with immunotherapies, for instance. However, some tumors have a paucity of anti-tumor immune cell infiltration and are much more immunosuppressive. These patients tend to have poor outcomes. To the end of converting responders to non-responders, we propose that focused ultrasound, specifically thermal ablation, can aid in achieving this end. To identify possible therapeutic combinations that synergize well with FUS thermal ablation, or TFUS, we performed serial bulk RNA sequencing on B16F10 melanoma tumors at 8, 24, 72, and 168 hours post-thermal ablation. First, we found that TFUS transiently controls tumor growth compared to sham treatments. We performed RNA sequencing on the tumors at the time points indicated by the black arrows. Second, we found that hundreds of transcripts are up or down regulated 8 hours post-TFUS. Some of the most notable ones are highlighted with green circles, and these include IL-33, IL-1 beta, HSPB1, and NLRP3. One of the reasons we highlighted these particular transcripts, among others, is that many of these transcripts are associated with the pyroptosis pathway and inflammasome formation. These are highlighted in the red boxes. So all along the pyroptosis pathway, we see transcript upregulation. However, we only see this temporarily because when we performed RNA sequencing at 24 hours post-TFUS, we see a relatively quiescent transcriptome, where only a couple dozen transcripts are up or down regulated. Since immune cells are known to be potent secretors of end products of pyroptosis, like IL-1 beta, we wanted to see if intratumoral immune cells were changing their IL-1 beta protein levels downstream of the transcript upregulation we previously saw. We are interested specifically in IL-1 beta because it acts in a multifaceted way to bridge innate and adaptive immunity. IL-1 beta can be pro or anti-tumor depending on the context. For instance, if secreted by dendritic cells through the NLRP3 inflammasome pathway, IL-1 beta is anti-tumor. However, if secreted by myeloid-derived suppressor cells, IL-1 beta is pro-tumor. And so to interrogate the IL-1 beta levels, where we previously used RNA sequencing, we performed flow cytometry on tumors 24 hours post-TFUS. This time point was chosen to give enough time for the translation of those transcripts to occur. And we found that the MF5 of IL-1 beta in CD45 positive IL-1 beta positive cells significantly increases, which means that there is more IL-1 beta per cell on average. To dissect this even more, we subdivided the immune cell compartment into a variety of phenotypes. The contribution of macrophages to IL-1 beta immune cell population significantly decreases 24 hours post-treatment. This carries over when we subdivide the macrophage compartment into M1-like or M2-like macrophages. This large decrease in representation also continues into monocytes and dendritic cells and CDC1s and CDC2 type dendritic cells. So the question is, from where is the IL-1 beta being produced? And the answer turns out to be granulocytes. In TFUS treated tumors, granulocytes make up the overwhelming majority of IL-1 beta positive cells in the immune compartment. So we see that the IL-1 beta protein levels are higher in immune cells, specifically granulocytes, 24 hours post-TFUS. Corollary to the question regarding immune cell IL-1 beta expression is if TFUS alters the intratumoral granulocyte representation. One helpful method to interrogate this question is by using dimensionality reduction techniques like T-distributed stochastic neighbor embedding, or TISNE. This provides an easy way to visualize changes in cell populations for subsequent downstream investigation. To this point, we see a massive difference in the center cell population, which is outlined in red. This cell population seems to be coming almost exclusively from TFUS tumors. When we overlay Ly6G expression, which is a classic marker for granulocytic immune cells in the myeloid compartment, we see this cluster is predominantly Ly6G positive, with Tisney suggesting we examine the presence of granulocytes. We performed classical hierarchical gating strategies and found that there is a huge difference in the proportion of granulocytes in TFUS-treated tumors, with a concomitant decrease in proportions in all other examined immune cell subsets 24 hours post-FUS. So next, with all of that considered, we decided to block NLRP3 inflammasome formation with a small molecule inhibitor, MCC950, in combination with TFUS treatment in vivo. We injected MCC950 intraperitoneally 1 hour before TFUS treatment. In this model, TFUS provides tumor control, but it is modest and not sustained. This is comparing the dark and the light blue lines. When we add MCC950, we see a substantial shift in the growth curve to the right. It is not quite statistically significant versus TFUS alone, but it does give a far greater degree of significance when compared to the sham curve. Again, this is only a single injection of the drug right before treatment, so optimizing the MCC950 dose has not yet been done. But in essence, it is consistent with the hypothesis that we had that inhibiting inflammasome formation will improve tumor control, perhaps by reducing IL-1 beta and granulocyte recruitment, although we would need to do more studies to determine that definitively. In conclusion, TFUS acutely upregulates pyroptosis and inflammasome pathways, and it also increases immune cell expression of IL-1 beta, specifically in granulocytes, and TFUS seems to increase the proportion of granulocytes in the tumor myeloid compartment. Finally, inhibition of NLRP3 synergizes with TFUS in transiently controlling melanoma tumor growth. Future directions, we plan on mapping the relationship between NLRP3 expression, ASC spec formation, which is involved in the inflammasome complex formation, and cytokine induction with respect to thermal ablation zones. We also plan on defining the role of tumor versus host NLRP3 inflammasome resistance to FUS, and we plan on testing whether the IL-1 family cytokines induced by TFUS serve to promote immunity or resistance. Finally, I'd like to thank a few people that were heavily involved in these projects, Alex Matthew, Lydia Petricka, and my advisor, Richard Price, and our collaborator, Dr. Timothy Bullock.